Explain how an increase in the secretion of renin stimulates the secretion of aldosterone. So this is the renin, angiotensin, aldosterone system. And so it's basically asking you, how do we get from renin to aldosterone? So remember, renin, one of those juxtaglomerular cells, specifically the granular cells, comes into contact with the inactivated form of angiotensinogen. That creates angiotensin 1. Angiotensin 1, under the influence of ACE, angiotensin converting enzyme, becomes angiotensin 2, and angiotensin 2 stimulates the adrenal cortex to make aldosterone. So we can kind of think of renin and aldosterone as synonymous, right? They have the same stimuli and they're kind of bringing about the same effect, right? Just that renin is doing so indirectly by activating aldosterone downstream. The second part of the question, how does an increase in aldosterone secretion affect sodium reabsorption? So aldosterone, we said, is going to reserve or pull back or reabsorb sodium. It's going to try to hold on to sodium, and it does so by inserting two things. It has that twofold effect. Inserting the leak channels into the apical membrane, and then also inserting more sodium potassium pumps in the basolateral membrane. And so by doing that, it's bringing sodium into the cell and then pumping sodium across. Okay, so it's going to facilitate more sodium reabsorption. Where are the cells that secrete renin located? So these are the juxtaglomerular apparatus cells, specifically the granular cells, which are located in that apparatus. Remember, we talked about the anatomical area in between the distal part of the tubule and in between those two arterioles, the afferent and the efferent, and that's where the juxtaglomerular apparatus is located, and that's where the cells that secrete renin are. Name 3 stimuli for renin. So the 3 stimuli that we mentioned, if we go back to this slide, decrease in the sodium and chloride in the distal part of the filtrate. And what this is really communicating is that there's a decreased GFR. The second stimulus was baroreceptor reflex activity, which is also communicating a decrease in pressure, right? Because we now have sympathetic, we now have decrease in the stretch of those arterioles because of the decrease in pressure. And then the third stimulus was decrease in afferent arterial pressure, which again is indicating to us a decrease in overall mean arterial pressure. So these are the 3 stimuli for renin, also listed here: decreased pressure, sympathetic activity, which is another way of saying decreased baroreceptor stretch, and then less sodium and chloride in the distal filtrate, which is communicating to us decrease in GFR. The third part of the question, how does atrial natriuretic peptide affect sodium reabsorption? So again, we're thinking about aldosterone and ANP as opposite ends of the spectrum, 2 sides of the coin, right? ANP is going to want to get rid of sodium. Aldosterone is going to want to hold on to sodium. So ANP is going to have the opposite effect. It's going to take out or block or inhibit those sodium leak channels in the apical surface of the membrane, the apical surface of the cells, which prevents sodium from being reabsorbed into the cell. It's going to have the opposite effect of aldosterone in an attempt to lose or waste or get rid of sodium in the urine. Okay, the third objective is to explain the role of aldosterone in potassium balance. So as we spoke about the mechanism of aldosterone, I kind of told you to stick a pin in its mechanism because it's also going to call on or involve potassium movement as well. But before we get into that, let's talk about some different potassium states. So there's hyperkalemia. This is a state of high plasma potassium, and hypokalemia, a state of low plasma potassium. And so plasma potassium, much like plasma sodium, needs to be very tightly regulated because of its crucial function to neurons, to muscle, and other excitable cells, okay? In fact, we cannot the body does not handle excess levels of potassium very well. There'll be lots of neurological consequences. There can even be death. There can even be fatality. In fact, the lethal injection is an excess dose of potassium. So that shows us how lethal hyperkalemia can be, okay? So we really want to regulate the amount of potassium in our plasma, and the kidneys, specifically aldosterone, plays an important role in doing that. So let's talk about how potassium is handled in the kidneys. It's freely filtered across the glomerulus. It is reabsorbed to some degree in the proximal tubules, although the mechanism there is not quite clear. And then in the distal tubules, it is reabsorbed and secreted, and this is regulated under the influence of aldosterone in the principal cells. So much like what we saw with sodium, it's going to be unregulated in the proximal part of the tubule and regulated in the distal part of the tubule. And then secretion is also going to take place in the distal part of the tubule. So aldosterone is going to have an influence on the principal cells, and it's going to be stimulated, it's going to be released in response to excess potassium in the filtrate. So if we have too much potassium in the filtrate, aldosterone, because its job is to get rid of potassium, it's going to be stimulated under those circumstances, and it's going to insert more of those potassium channels, which will facilitate more potassium secretion. So we can think about aldosterone as having opposite effects when it comes to sodium and potassium. Aldosterone is going to want to hold on to sodium and get rid of potassium. So here is the mechanism for reabsorption in the proximal tubule. Again, the way that it moves through the cell is not quite clear. This mechanism is unknown because if we think about the gradient here, the sodium-potassium pump is pumping potassium into the cell and sodium out of the cell. So we have a higher concentration of potassium in the cell. And so there is no passive gradient for potassium to leak. In fact, potassium is leaking in the opposite direction. So the mechanism of how it gets into the cell is not quite understood, but we know that there is a bit of paracellular transport happening because of the leaky nature of the tight junctions here. And so potassium is thought to be moving in between the cells in the proximal tubule where we have those leaky tight junctions. In the distal part of the tubule, the mechanism is quite clear, right? We insert more leak channels in the apical surface. This is how we allow potassium to leak into the tubule, and this is how potassium is secreted. So it's being pumped into the cell across the basolateral surface by the sodium-potassium pump, and then it's leaking in the opposite direction via the leak channels which are being inserted in the membrane. And if you remember earlier, I said aldosterone is inserting two channels in the membrane of these principal cells. It's inserting sodium channels, which leak sodium into the cell, and potassium channels, which leak potassium in the opposite direction. Okay. So let's talk about this question here. Which hormone regulates potassium secretion? So aldosterone, we just talked about that. What are some stimuli for the release of aldosterone, of this hormone? So the stimuli for aldosterone, again, I'm gonna kind of encourage you to think about renin and aldosterone as having the same stimuli. Because remember, renin is what eventually becomes or creates or stimulates aldosterone. So they have the same stimuli. And the stimuli are increase in sodium and chloride in the filtrate, increase in the baroreceptor, increase in the sympathetic activity, which is decrease in the baroreceptor stretch in arterioles, and then a decrease in the afferent arterial pressure. So here, these three stimuli are going to be the stimuli for renin, but we can also think of them as being the stimuli for aldosterone as well, because as we know, renin becomes or begets aldosterone, alrighty? And then the fourth one we want to add in here is how it pertains to potassium. So whereas decreased levels of sodium in the filtrate stimulate aldosterone by way of renin, increased levels of potassium in the filtrate stimulate aldosterone as well. Okay. All right. Okay, so let's move along to the final renal packet. And typically I cover the acid-base homeostasis of the respiratory and urinary systems together, but I think you already covered this with Dr. Schlater, the respiratory component of acid-base. So you're gonna wanna eliminate or ignore the entire first part of this packet. So I believe slides slides 1 through 9. Yeah, so you would have covered this with Dr. Schlater, which is the respiratory component of acid base, and we're gonna pick up with the end of the packet, which is the urinary component of acid base, okay? All right. So here we're talking about how the kidneys play a role in the acid-base balance. And you should already be familiar about acid and base and the role of the lungs in all of this, but we're gonna pick up here and speak about how the kidneys also play a more long-term regulatory role in our acid-base balance. What we saw with the lungs is that the lungs have an immediate influence on acid base in the form of our respiratory function and hypoventilation. The lungs can easily get rid of excess acid by blowing off excess CO2. The kidneys also have a way of regulating acid base, but in a more long-term way. It's not going to be as immediate as what we saw with the lungs, but it will be just as beneficial or influential in terms of our overall plasma pH. So let's start here by looking at the interaction between fluid and electrolyte balance. So the increase in solute reabsorption is what influences our osmotic gradient for water reabsorption. And this is the whole notion that we've kind of been slowly describing so far. When we reabsorb sodium, we create a gradient for water to follow, right? Water follows sodium movement. And so when we look at ADH and its role in correcting our fluid and electrolyte balance, ADH primarily has an influence on water reabsorption. And just to jog your memory here, that's going to be the way ADH influences the insertion of those aquaporin 2 channels that we talked about in the apical membrane. But, and I put also in brackets here just to kind of clear this up, ADH also has an influence on sodium channels, all right? And inserting more sodium channels into the apical membrane, hence reabsorbing more sodium. So while ADH has a main role of reabsorbing water, it also has a secondary role of reabsorbing sodium. And the same is true for angiotensin II. Angiotensin II has a main role of reabsorbing sodium by inserting the leak channels, inserting more sodium potassium pumps, but it also has a secondary role of increasing ADH secretion, which is going to reabsorb more water. So its primary role is going to be reabsorbing sodium, but then its secondary role is going to be reabsorbing water. And then ANP has a similar phenomenon. ANP had a main role of excreting more sodium by blocking reabsorption. And its secondary role is going to be decreasing ADH secretion, so blocking water reabsorption, which is to say that it stops the insertion of those aquaporin 2 channels and prevents more water reabsorption. So I don't want you to be confused about the terminology on this slide, but it's just basically showing that all of these hormones kind of tie together hand in hand. They're basically trying to bring about one goal. So in the face of, let's say, hemorrhage or hypotension or shock or dehydration, ADH is going to try to reabsorb more water, but it's going to have a secondary role of reabsorbing more sodium. Both of those effects are going to be in an attempt to correct that initial imbalance. And the same is true for angiotensin II and ANP. Okay, so while we described them earlier as having a primary role, please understand that they do tie into the secondary role, all in an attempt to fix those initial imbalances. And so that's what we mean by fluid and electrolyte balance being kind of hand in hand. Anything that's trying to correct fluid balance is also attempting to correct electrolyte balance, sodium, and anything that's trying to correct electrolyte balance also is going to have a role on fluid balance as well. Okay, now the physiological response that we see in terms of hemorrhage is a decrease in blood volume, which translates to a decrease in mean arterial pressure. And the neural control to the heart is going to have an effect on the baroreceptors, and that's going to decrease their stretch, which increases sympathetic activity. And then the hormones that play a role are ADH and renin, angiotensin, and aldosterone. Okay, so these two influences, or these three influences rather, are trying to correct that initial imbalance, that initial decrease in blood pressure, or that initial shock. So that's ADH, that's the renin, angiotensin aldosterone system, and that's the baroreceptor reflex. And just to connect this to what we just talked about, these are the exact same stimuli for renin in the face of hypotension, shock, or hemorrhage. Okay, let's move on to some of the other objectives here. We're going to describe the various factors that influence acid-base balance, and then we're going to talk about how buffers in the blood, the action on the kidneys per se, and then we'll kind of touch on what's happening in the lungs, although you would have talked about that already. So in terms of kind of going over the basics here, we know that our normal arterial blood has a pH of anywhere from 7.35 to 7.45, so a very narrow window of regulation for our blood pH. Anything that goes less than that, anything less than 7.35 is acidosis. Anything greater than 7.45 is alkalosis. And remember, acidosis is excess acid, alkalosis is excess base. Now, the complications that can be possible with some of these acid-base disturbances are configurational or conformational change in the structure of our proteins. Remember, proteins have that primary, secondary, tertiary structure, and that secondary structure is very sensitive to the environment of the proteins. Heat or acidic or basic environments can denature these proteins, can cause them to misshape or misfold, and that's going to obviously have a functional disturbance in the way that those proteins behave and function. So that's an effect that we don't want, which is why we try to keep our pH in that window. Other effects are changes in the excitability of neurons. So if there's an acidotic environment, neurons are less excitable. It's going to take longer. It's going to take more. It's going to be harder to fire action potentials. Whereas if we have an alkalotic state, neurons are going to be more excitable. We're going to be very sensitive. This is going to be like spasticity. Neurons are going to fire action potentials much more easier, right? Much more easily. And so changes in potassium balance can also be influenced by these acidotic and alkalotic states. If we have excess acid, we have hyperkalemia because hydrogen ions tend to attract potassium with them as well. So anywhere there's excess acid, excess hydrogen, we also see excess potassium, and the opposite is true. If we have too little hydrogen ions in the form of alkalosis, we begin to see hypokalemia as well. And I talked about why we want to keep potassium in a very tight balance. Also, because of its involvement in excitable cells and because excess potassium can actually be fatal. So it can be fatal in causing cardiac arrhythmias, and it can be fatal in causing vasodilation as well. Now, just to kind of give you more of a practical application here, some of the things that we typically see in an acidotic patient is fatigability, just probably fainting, just depression of the nervous system in general. Where some of the features that we see in alkalotic patients is excitability, sometimes nausea, sometimes vomiting, spasticity of the muscles are some of the symptoms that we see in alkalotic patients. So let's talk about the sources of input and output for our hydrogen ions. So hydrogen comes in through our proteins, through fats. It's also a part of the metabolic byproducts of some of our, so for example, muscle, if we're breaking down glucose, right? Lactic acid is produced. Keto acids are produced depending on the source of that energy. Carbon dioxide is produced as a byproduct of cellular respiration, excuse me. And so all of these are sources of hydrogen ions into our blood. So in an attempt to prevent excess hydrogens, we need to have other outputs that can balance this excess acid. And those are the kidneys and the lungs. So the lungs can balance acid by way of CO2 production, and the kidneys can balance acid by way of secreting or getting rid of, excreting excess hydrogen ions. And this is another reason why excess proteins can be taxing to the kidneys, right? We kind of alluded to proteins and the fact that proteins break down to urea earlier, not urea, excuse me, it's break down to nitrogen earlier, but this is another mechanism as well. Proteins produce, when they are broken down, produce excess acid, and that can create an acidic environment in the blood. Fats as well. So the respiratory disturbances, let's talk about those here a little bit. Now, carbon dioxide, we know, is a biological acid. It behaves as an acid in our body. And so the way that the kidneys, the lungs can convert that CO2 back to acid is via this enzyme carbonic anhydrase. So the combination of CO2 and water and the enzyme carbonic anhydrase can create carbonic acid, and then carbonic acid can freely dissociate into bicarbonate and acid. And so this is how we kind of move acid around in our blood in the form of CO2, and then when it gets to the lungs, it can be converted into acid and vice versa. Now, the normal PCO2 in our blood should be about 40 millimeters of mercury, and this is maintained by the respiratory chemoreceptors in our brain. And so the brain is basically detecting the acidity of our blood by determining what the arterial PCO2 is. Some of the sources of CO2, as I mentioned earlier, metabolism of fats, proteins, lactic acid production by muscle, and so on. And some of the outputs I mentioned earlier as well are through the respiratory system by blowing off CO2. And so if we have increased plasma CO2, that can create a state of respiratory acidosis. If we have decreased plasma CO2, that is a state of respiratory alkalosis. Now, the main takeaway here is that the respiratory disturbances are caused by the lungs. So if we see the word respiratory in front of this disturbance, we know that the initial imbalance is happening in the lungs. And so if the initial imbalance is happening in the lungs, that is going to influence the plasma CO2 directly. Let's talk about some of the other sources of acid-base disturbances. So these are the metabolic disturbances. Now, again, if we see the word metabolic in front of the disturbance, whether it's acidosis or alkalosis, we know that the disturbance is being caused in the kidneys, okay? So this is a gain of hydrogen ions or a loss of bicarbonate ions that is really originating in the kidneys. So a decrease in pH through something other than CO2, something other than the lungs. So this can be a high protein diet. This can be a high fat diet. This can be heavy exercise, which produces lots of lactic acid. This can be severe diarrhea. If we lose a lot of fluid in the bowel, we also lose a lot of bicarbonate with it. And if we lose bicarbonate, we're creating a state of acidosis. Or this can be renal dysfunction. So any cause other than what's happening in the lungs, so either in the kidneys or excess diet or exercise, we're going to call this a metabolic acidosis, okay? So a gain of hydrogen or an excess loss of bicarbonate is going to put us in a metabolic acidotic state. Now, we can also be in the opposite scenario. This is a metabolic alkalosis. This is a state of excess loss of hydrogen ions or excess gain of bicarbonate that is created in some way other than through the lungs, other than via CO2. This can be caused by excess vomiting. Remember, the stomach has excess hydrochloric acid. And if we lose that hydrochloric acid by vomiting, then we're losing hydrogen ions and we're throwing ourselves into an alkalotic state. Also, the consumption of alkaline products like baking soda can produce an alkalotic state. And then the kidneys as well, if they are losing excess hydrogen ion, secreting it in the urine, or holding on to or reabsorbing excess bicarb, that is going to put us in a metabolic alkalosis as well. Let's look at the 3 lines of defense here. So the body has 3 lines of trying to defend us against some of these acid-base disturbances. The first line is called the buffering of hydrogen ions, or the buffering system. So a buffer is anything that tries to resist a change in pH. And this can only prevent pH disturbances. It cannot reverse or correct pH disturbances. And we talked about one of the main, or maybe we did not, we'll probably talk about it in a few slides down, but the main buffer in the body is going to be our bicarbonate, right? It's going to be able to create carbonic acid as a way to buffer the changes that would otherwise be experienced if we had just bicarb or acid floating around. And so it's going to create carbonic acid, and that is going to help buffer any changes in pH. That's the first mechanism, buffering of hydrogen ions. The other 2 mechanisms are respiratory compensation and renal compensation. Now, these 2 mechanisms can actually reverse or correct changes that may be experienced in our pH or disturbances in the pH. The respiratory compensation is going to be a homeostatic regulatory mechanism. It's going to be kind of an inbuilt mechanism in the body. So if you think about your rate of breathing, whether you're hyper or hypoventilating, that is in response to those receptors in the brain that are detecting any change in your PCO2 in the blood. So that's the respiratory compensation. So let's say you're in a state of metabolic alkalosis, right? Let's say you had an excess diet of proteins. Let's say you had vomiting, right? You lost a bunch of acid and now you're in a state of metabolic alkalosis. The kidneys can kick in and try to compensate that disturbance by decreasing your ventilation, decreasing your rate of ventilation, putting you in a state of hypoventilation in an attempt to hold on to more CO2 to correct that initial alkalosis, right? And that's going to kind of balance out your pH. Now, as I mentioned earlier, the respiratory compensation is going to be more immediate because I can sort of on a whim change my rate of ventilation based upon what my those centers in my brain are picking up in my PCO2. The renal compensation, on the other hand, is going to be much more of a long-term compensation. Here, we can